This recorded session is dedicated as our course orientation for S-Biol 418. The subject is Cell and Molecular Biology lecture for three sections, MEB 3.1, MEB 3.2, and MEB 3.3. I am Arizaldo Castro and I will be your course instructor for the semester. We meet the following schedules for MEB 3.1 every Wednesdays, 10:30 to 1:30. For MEB 3.2, we meet on Tuesdays and Fridays. Uh, we meet at 2 o'clock to 3:30. And for MEB 3.3, Tuesdays and Fridays as well, we meet at 3:45 to 5:15 p.m. So for this course orientation, I'll be discussing a few things about the course syllabus, the topics, our grading system, the implementing guidelines that we are going to follow in the distance learning setup, since every interaction, output, and assessments that you will be taking or that you will be making for the subject will be given in the virtual platform that we are using. So everything will actually be um, done using our schoolbook platform, our schoolbook e-class. Um, then lastly, I'll talk about or I'll also discuss our procedures for engagement. So whenever you express your concerns, your question, or if you have clarifications or questions about the about certain topics of the course or the whole course itself. So let's begin with the course overview. So this course will cover discussions about the structural features of cells. We'll dive will dive deep into studying ultrastructures to the point that we are going to dedicate specific modules for the organelles of the eukaryotic cell. But before we dig on those topics, which are very specific to the cell ultrastructures, we're going to cover some fundamental discussions about the chemistry of the cell. So we'll go back to the basics, things that probably you've learned in your organic chemistry or your biochemistry classes. We're going to review them, and then that will serve as our preliminary topics to enable your learning or to enable your learning path dun sa pag-discuss natin on each of the organelles of the eukaryotic cell. Also, we'll at the end part on the finals, more on the final modules, we will cover the principles of central dogma. So how information is transferred from one biomolecule to another biomolecule. So you will probably recall the terms DNA replication, transcription, and translation. Those are things that we will cover in depth at the final module, the final part of the module. And then we'll talk about how the process of um, uh, information flow from one biomolecule to another actually influences the homeostasis or the stability of at the cell level, at the cellular level. We'll look into how the ultrastructures and information flow, how these two important themes in our lecture, how are they how can we use them as narratives to understand cellular functions and cellular stability. The subject will be delivered using the distance learning setup, so there will be no face-to-face classes. Everything that we are going to every material that I'm going to provide, every output and assessment that you are going to produce and take All of them will be conducted using the Schoolbook platform. So with that, we made sure that the flow of topics in the curated content will follow the two frameworks that are applicable for higher education subjects. So we made sure that the subject is compliant to the outcomes-based education framework and also the 21st century learning development frameworks. Frameworks. So we made sure of that the course delivery is standard among all sections. So we can still foster and we can still foster learning, no? Even though we are on a very very unique setup that we're not having in-room classes. So much about the course and the modality, the pedagogy that we are going to employ. Now let's have the next section. So a bit of information about myself as your instructor. I also graduated from our university. I took my bachelor's and my master's degree in our university, and right now I'm finishing my doctor in philosophy and biology at the UP Diliman Institute of Bio. I was able to work for our university for straight years after I graduated as a lecturer. And then afterwards, I worked for the Department of Health as a research fellow. Then I returned to our university last year as a faculty teaching the health sciences course, the health sciences subject, especially those that are in the fields of microbiology, molecular biology, and immunology. My research interests as an academic include are centered on microbiology, in particularly microbial ecology of aquatic systems. I'm very much interested in learning, in elucidating the phenomenon of antimicrobial resistance in built environments as well as in water systems. I'm equipped with training, both graduate and professional training on bioinformatics and molecular phylogenetics. So essentially, my strengths are on the molecular sciences. Then during my master's or during my master's degree, my master's training, no, a lot of specialty courses that I took revolved on systems bio, particularly animal models. So I teach I teach how molecular principles and themes apply to the health of vertebrate organisms particularly in humans. No? Then, lastly, I'm also interested in doing impact tracing work, impact assessment work, which is something new in our country. So, uh, things that or most of the activities that are related to impact assessment most of the activities that I've done related to impact assessment were actually during my stint with the Department of Health. So right now, I'm working as a part-time faculty in the department. I started last second semester, so I think I handled some of your batchmates or some of the. I think I handled MEB 2-3 already. So the section should know me, or the students should be able to recognize me for this semester. Okay, now next, the requirements that we have. Grading system that we are following is that we're going to follow is the absolute grading system. No transmutation of any sort. So whatever you get will be computed as is. So if you, let's say you have an enabling assessment that is worth 10 points, you get 5 points out of it, out of 10, then your score will be 50%. Okay? Last semester, our grading system for all subjects is set at semestral grade. Semestral grade will be equivalent to the average of your midterm and your final grade. And for each term, there are only two components. 60% will be dedicated to enabling assessments, while 40% will be for summative assessments. So some revisions were made after the IRR organizing or revisioning revision committee, IRR organizing or revision committee uh, um, consulted with the different members of the academic community. So we're not going to follow that old grade system anymore. We dedicated 15% for class participation, and we took that from the 60% of the enabling assessment. So your enabling assessments will now be graded as 45% of your term grade, and the 15% will be for class participation. So what is class participation composed of? A major chunk of the class participation are your attendance to synchronous meetings. So I know it might sound, challenging. It might sound disappointing also, since for the longest time you are not required to attend synchronous meetings because we're recording them, and the IRR says that of course we have to consider different conditions, and I acknowledge that. But the thing is, with the um, suggestions or with the consensus of the community members, that this new rule will follow for this academic year. So what I can do, what I thought of course, to balance that, because I still acknowledge the fact that not all of you will have the same level of access to no no to online resources every time. So what I will plan to do is, aside from the attendance, I will also get a portion of your class participation uh, um, via outputs, no submission of outputs. Like I can ask you to answer a forum question, discuss your thoughts about something, about an article that I asked you to read, and then you voice your thoughts about it, or your answers to the question I asked, and I will tag that as class participation. So for me, it's not going to be purely attendance. I will be tagging some of your outputs as class participation. Okay. So same thing for enabling assessments and summative assessments. They still will be a mix of different exam types. Could be a mix of identification, true or false, enumeration, problem solving, or case analysis. And it will vary from time to time, depending on the module content, depending on what I think will be the best to assess your competency for a specific module. Next part, exam types. So there will be an individual, or I can give exams individually or by group, no? For enabling assessments. Usually for enabling assessments, saan tayo pwedeng mag-group, no? Aside from those graded assessments, that might be quizzes, discussion papers, I also give formative assessments. So these formative assessments, they are ungraded, so they will not be counted to against your semestral or your term grade. And some of them include reading assignments, offline reading assignments, like you're tasked to read a book chapter, a journal article, an online web page, or an online wiki page, no? And then you will tag yourself as completed on that particular component. That's formative. That's a formative assessment. Also, I may ask you to screen a video, a short video, or a film, and that is also considered as a formative assessment. And there are many other types of formative assessments that I can give all throughout the semester. For enabling assessments, which can be individual or by group, no, usually quizzes sila, mix of different questions. And I can give, for enabling assessments, at the least one or maximum of two per module, but probably I'll stick to about one enabling assessment per module to, of course, minimize the strain on your end. But please expect that since this is a specialty subject, it's not like general physiology or general microbiology. No, this is a I don't know a 300 or 300 400 level subject. We have to, or my approach will be quite different. No, that the exams will really be a mix, a hybrid of objective and um, a hybrid of objective and what do you call this? Uh, Analytical, uh, hybrid of objective and analytical exams. No, you expect that aside from the objective questions, I will include case analysis or problem solving. No, because we're not only studying theory here. We are also going to make little mentions about instrumentation, about uh, specific, research techniques. And you actually have a co-requisite for the course, no? Meron tayong cell and molecular biology lab. So in a way, we will also try to incorporate the laboratory competency with the theory. Now, for summative assessments, these are graded exams. There are a total of 4 exams all throughout the semester. There are 2 summative assessments for the midterm and 2 for the final. And summative assessments are not entirely different with enabling assessments, but of course, the questions and the approach will be a little difficult, no? A level higher compared to your ordinary quizzes, because summative assessments are like your prelim exams, your midterm exams, your summary exams that will really test your competency, your overall competency for the specific term. Okay, so enough about the course requirements on exam types. We'll now cover the course outline, the things that we will be discussing in the lecture. So there are 12 modules. The first module is dedicated as the course orientation week. So this recorded lecture, recorded session is actually part of module number 1. So after this week, we're going to proceed to module number 2, which will be all about the cell. We'll take a look at the cell and identify or study history, a little bit of history on the study of cell biology and also molecular bio. We'll look into disciplines that are related to CMB and how these disciplines contributed to cell and molecular biology. We'll also talk about an overview on the cell. We'll cover discussions about, or we'll have discussions about the morphology and the what do you call this? The morphology and physiology, little bit overview lang naman, of prokaryotic cells and eukaryotic cells. No? So since this is cell biology, we are not going to make any distinction between bacterial cells, archaeal cells, and I'm not saying that. Sorry, let me rephrase. We're not going to focus only on a specific type of cell. So our discussions, the narratives, what do you call this, the theory that I will be providing, I will be discussing throughout the semester, will be both covering, will cover both prokaryotic cells and eukaryotic cells. Okay, so for module 2, fundamental discussion on history and the structural biology, as well as overview on physiology of eukaryotic and prokaryotic cells. The next module will cover, will have a review of your organic chemistry or your biochemistry. We'll talk about the chemistry of cells. And we will zoom in to discussions about the carbon atom. As you know, it's the central atom of organic molecules. It's the most important atom of living organisms. So we'll have the spotlight to the carbon atom. Also, we'll talk about water. We'll discuss its properties and the repercussions of these properties in the maintenance of a stable or a steady state in cells. No? Also, we'll talk about macromolecules and polymerization, no? For the next module, since these are actually these two modules, chemistry of cells and macromolecules and polymerization. They're very connected, or they're very important for us to understand further yung ating mga enabling topics. No? So this might be a review or something totally new to you. I'm not very sure because with regards to your organic chemistry or biochemistry, I'm not sure how the curated content that were provided or that were given to you as material, as learning material. So for macromolecules and polymerization, this module will be dedicated to the discussion of the major biomolecules of living organisms. So we'll cover an introduction or an overview of the carbohydrates, the lipids, proteins, and nucleic acids. We'll discuss their chemical properties, their structure, and some examples. We'll provide examples on how their properties actually influence or how their properties dictate their function in a specific cell. No? Then, another important topic is polymerization. So, as a phenomenon that makes organic molecules highly structured from a simple from the first level to the highest level, polymerization is very important, and it's something that we have to cover because I'm not sure if you covered it in your biochemistry or your organic chemistry. So for our course, we will discuss in depth the process of polymerization, its kinetics, the physicalities of polymerization. We will cover that because eventually eventually our knowledge of these two things about the structure and the way macromolecules organize themselves will be important in our succeeding topics in the discussion on ultrastructure and organelles and the functions that they carry out. Okay, so enough of the enabling topics. We'll now, the preliminary topics. We will proceed to the enabling lectures. So for the enabling lecture, we will start this with a discussion on the cell membrane or the membrane, because it's not only the structure of the structure of cell membranes, not only the one that you can see outside or the one that demarcates the proximity the area of the cell. No, so membranes are also found within cells. Diba? We have our membranous organelles. The membranes of these organelles are actually of the same chemical nature and chemical organization as the cell membrane that is covering or demarcating the boundaries of the living cell. We're going to cover a discussion on the history of how the cell membrane was studied, the important characteristics of the cell membrane, the different theories that are proposed about that try to discuss the nature and biology of cell membranes. So aside from that, we're also going to include our discussion on cellular transport. So cell transport is very important because for interactions to happen, there should be mobility of entities, cellular and subcellular. No? So cell transport will cover active transport and passive transport processes. So we will discuss chemical components of the membranes that actually facilitate the process of molecule transport. We will also talk about mechanisms, yung ano-ano, active and passive. We'll differentiate them. We'll talk about some examples of active and passive transport mechanisms. No? So this might be a review. We will review yung mga ano nyo, basic understanding nyo about cell transport from your Bio 100. And we are going to reinforce those learnings with how cellular transport, how important it is for the homeostasis of cells. Then, another layer of cellular transport or another department that we're going to cover is the endomembrane department. No? So endomembrane system is a set of organelles within the eukaryotic cell that are behind protein transport, protein synthesis and protein transport. So we're going to discuss them in our lecture, the organelles comprising the endomembrane system. In addition, we will also talk about cytoskeleton as a sort of skeleton, no? Or what do you call this? Scaffold, kumbaga, ng cell. No? So we'll talk about cytoskeletal filaments and their process, the process in which they form, how they function within the cell, and some important narratives on specific molecules, no, specific. Actually, not specific, but example molecules that are of cytoskeletal nature, and then how they function or their importance. We'll cover them in the lecture. And then we will look at the cytoskeleton as the primary enabler of cellular events such as cell division, motility. No? In this specific module, you will appreciate how the cytoskeletal or how the cytoskeleton actually plays a role in dynamic cellular events like cell division and other more activities that involve cellular motility or cell component or mobility of cellular components. Okay, so aside from the endomembrane organelles, the cytoskeleton, as few of those organelles that we're going to talk about, we are also going to talk about the chloroplast. No? So found in plant cells, in photosynthetic organisms, chloroplast will be one of the central organelles that we will cover in the enabling modules. We'll describe the anatomy of the chloroplast. We'll discuss photosystems and how we can relate the organelle structure of chloroplast with the process of photosynthesis. So we'll cover photosynthesis for not that in depth, but we will include that in our lecture discussions. The next set of module, or organelle, the next organelle we're going to cover for the enabling discussions is the mitochondrion. So the mitochondria, we'll talk about the anatomy, the organelle anatomy of mitochondria, and we will relate that with the process of cell respiration. So how the processes or the stages of cell respiration, how is it facilitated by the mitochondria? Then the dedicated module will be for Krebs cycle and the electron transport chain. So this might be something that is, or this might be a revisiting on your end, but please expect that we are not going to cover fundamentals of the Krebs cycle and the electron transport chain anymore. We're going into detail about the processes, the sub-processes within these processes, yung mga sub-steps or activities that are occurring, allowing them to proceed as expected. We're going to take a look at each of those steps, at each of those sub-processes, and we're going to discuss the cell biology, the molecular biology on such steps, on all of those steps. Next, the next set of organelle, or the next organelle, is the nucleus. Okay, we're going to cover also a discussion on the DNA. We'll describe the relationship between the nucleus as an organelle encompassing or enveloping the cellular DNA. No? And we'll talk about DNA organization at this module, so that we'll appreciate how um, organized it is, how is it able to retain so much information, no? Given that it only covers little physical space. Okay, and for the structure of DNA, we will be looking at the history of how it was discovered, how was it studied, how is it being studied to date, and we're going to take a look as well at the chemical properties, the chemical properties of the DNA. Um, I think after the topics that I've mentioned, we're now proceeding to the final or culminating topics. For the culminating topics, we'll focus on information flow, on lateral information flow. So from your DNA to your RNA to your proteins, we're going to take a look or take a deep dive into the central dogma of molecular biology. So we are going to discuss about nucleic acid replication and gene expression, protein synthesis. No? We are going to use two model systems, yung ating prokaryotic and eukaryotic. So in both of these model systems, we are going to discuss the three major processes: replication, transcription, and translation. We're all going to study them using two model organisms, the bacterial cell and your usual eukaryotic cell. Now, with everything that we have studied so far, from the chemistry of the cell to specific organelles to biomolecules, especially we're putting the spotlight on DNA. Another final topic that we will include in our lecture is signal transduction. No? So we're going to talk about the process of signal transduction. We will focus on specific molecules such as G protein link receptors and cyclic AMP, because these two are things that you will encounter over and over again whenever you study something that involves signal transduction at the cellular level. You will always encounter these two things. So we're going to give the spotlight to them. Also, we are going to talk about the adenyl cyclase, and very important enzyme, a very important protein during in different signal pathways. No? And at the same time, at the same level, we'll also talk about protein kinases. We'll have model signal transmission pathways. So we'll have some examples, and then we'll use them as our model examples. And we'll study them based on components, based on where these elements are involved, and ultimately, what's the output? What happens if that signal transduction pathway is activated or deactivated? Then, last topics will cover cellular cycle and cell division. We will describe the process that are the processes of meiosis, mitosis. We will look at how the process occurs, like flesh out natin what elements are involved, what signals are needed in order for division to take place, and then we will relate cell division to the normal functioning of the cell. And at the other spectrum, at the other end of the spectrum, we have abnormal functioning. So as a case, we will dedicate a specific module for cancer cells. So we'll discuss the different factors that affect normal activities of cells that may, contribute to the formation of cancer, to the development of cancer. And we'll also talk about self-healing cells and how, cancer cells will how cancer cells will influence the physiology of a normal cell that transforms as a cancer cell, and neighboring cells as well, because as you know, one of the hallmark characteristics of cancer cells is that they can modify the physiology and the behavior of the cells that surround them. Okay, so I hope that we are able to see the cycle, or the how the topics were arranged in such a manner that you will have, at the end, you will have appreciation of cellular stability or instability, and you will have the tools necessary to discuss such two opposite what do you call this, opposite things, no? So in the light of or using the lenses of uh, cellular chemistry to cellular organelles that are maintaining the homeostasis of the cell, to lateral information flow. So all these things as fundamental to understanding cellular health or cellular disbalance or cellular instability. So I hope that by the end of the lecture, that's something that you guys will be able to appreciate now. So there could be a lot of expectations right now, especially that at this juncture, ang daming topics that might make you feel overwhelmed. But I'll try my best to deliver them in such a manner that we will gain, or you guys will gain, more appreciation of the theory and the repercussions of the facts from the theory, or the facts that are tied by the theory. Okay? So for your expectations, please feel free to let me know of your expectations through the self-introduction output. So under Module 1, there is an output that I ask you to produce. So feel free to put that to right away provide any expectation, or articulate any of your expectations. All right, so we're now nearing the end of this course orientation recording. For course policies, I want you to see the updated syllabus, the uploaded syllabus rather. We there are specific course policies that I need to emphasize. First is on enrollment. If you notice, I did not add you personally dun sa ating CILP course. Actually, the initial course that you were enrolled in, they were CILP created courses. So I did not create them. So the reason behind that is because our university wanted to automate the enrollment process, such that only officially enrolled students can get access to the e-class and can participate in the discussions. So they automated the process. They did not added that, or they did not make us do that task anymore. No, so if you will be able to answer the transition survey I added on that e-class, you will be enrolled to our official e-class. Okay? So again, just to reiterate, I don't have any capacity to enroll you manually in our e-class. It's the CILP and registrar's work. So you cannot message me, or you may message me, but I will give you the same response. I don't have the capacity, and you guys as students also will have no capacity to enroll your classmates, or if you, even though you give the access code or whatever information linking or identifying our official e-class, your classmates still will not be able to enroll unless they are added automatically. Okay? Next is policies on communication and feedback. So, in terms of our interaction, well, we communicate during synchronous meetings. We can also do online question and answer from time to time. Some of you, since some of you were my students before, probably we already have our ano naman, no? Our working dynamics on how you contact me, how I communicate with you. So you can contact me anytime, but please expect that I will not respond in real time, especially if you contacted me during ungodly hours. No, so but of course on my end I try to really reply real time, especially during work hours. Okay, for attendance, yun, yeah, since we are covering, we have our class participation as a component, so we're going to include uh, attendance to lectures, to synchronous meetings as your as part of your term grades, no? So for attendance naman, what do you call this? I won't be strict on how you will appear during the synchronous meetings. By the way, I will be including, or I will be requesting that you turn on your cameras for the first 5 minutes of our lecture, so I can verify that it's really you that is listening to the lecture. No? And then afterwards, you can turn it off. During my lecture, it will be actually friendlier if you will turn it off. No, so I have to add that, no, that you need to open your cameras for a while for me to validate your attendance. Then for intellectual property, we encourage the students to use original material, or if they lift from a reference, they have to properly cite it. So on my end, same policy. Other policies, what else? On what do you call this? On your attire, I think other policies include like your attire during e-class, um, the way or how you will conduct yourself. No, those are things that those are things that I expect you to know at this point. Okay. Our medium of instruction is English, but I will do code switching from time to time to, put emphasis on certain things, on certain topics. But mostly, I will deliver the lectures in English. Even the recorded ones, like for our asynchronous meetings, the recorded lectures will be delivered or will be recorded in English. Okay? Now, my personal reminders and regulations that I want you to know. So I start the class on time. MEB 33 knows that. No, they were my former students in general microbiology, so they know that I start the class on time. Though I provide some leeway for students, no, about 5 minutes, I will allow students to populate muna in the chat or in the MS Teams before I start the lecture. And I mentioned this already, have to open your camera for attendance verification. Now, since this is a lecture class, no, I will enforce the idea, or I will, bring this idea about authentic learning. So, usually in face-to-face settings, um, or during my, ano naman, no, my previous experience with lectures, with my students, with my former students, I think the best method that I have used in order to elicit interaction and to elicit curiosity and desire for learning in my students is I ask them questions, I ask them critical random questions related to the topic, and I try to get answers from 2 or 3 students. No? So, before I really enjoyed those times because I can really sense that my students are becoming interested on the topic at hand. So for this setting, this is really challenging so far. But just like to remind you that if you will be able to participate actively, you'll able to engage yourself uh, in each module. I can assure you that you will really learn a thing or two. I cannot assure you that you will learn everything, but I'm sure that you will learn something from each module if you will take time to read the materials, to do advanced discussions among yourselves, or do advanced readings before the synchronous meetings. Because there are things that, of course, I will not be able to cover, things that probably you did advanced reading of, and some things that and probably I'm not aware of. And we can include that during the lecture discussions. And that will be beneficial not only to you, but also to your classmates. And third personal rule is I will not tolerate any act of bullying in my class. So everyone is expected to conduct themselves properly, especially during synchronous meetings. Okay? Now, because of our setup, I will have no means of telling or identifying whether someone is being bullied or not. No? Walang actual observation that I will have. So on my end, what I can offer to you is if ever that you feel that you're being outcast or bullied by your classmates, no, I'm the number one person that you can I'm one of the individuals that you can talk to so that I can offer help. I can offer help and we can resolve the issue um, as soon as possible. Okay, so please do not, I may look strict. I look strict and unapproachable during lecture meetings, probably. I'm not sure, but with regards to personal matters like this, you can trust that you can discuss to me, and I will offer my help, and we can probably bring this to a higher discussion to higher offices so we can resolve issues like this, issues like bullying. Another issue that I have personal stance against is, of course, I don't know, I think hindi lang naman ako, but for me, this is personally very important. Academic dishonesty. I won't tolerate any act of validated academic dishonesty. Right now, we don't have a very clear definition of what constitutes cheating, what constitutes academic dishonesty. For a highly technical subject, or for a theory subject, no, uh, usually in the face-to-face setting, this is not a rare thing. Cheating is not a rare thing. No, I've had my share of students cheating in my theory subjects, and mind you, those students, no, they either that they did not pass the subject or they did not graduate on time because I have personal commitment towards you know towards fighting this fighting cheating. No, I don't tolerate it because it's not proper in a setting that is in a in a uh, uh, university course setting. It's not tolerable. No? So in a setting where you are being graded for the competency that is being measured, okay, any act of cheating or any act of trying to game the trying to game how the scores will play out, how the scores will look like in the end. No? Those things that constitute academic dishonesty, they're not they will thank you for a lifetime. So I guess that's my moral plea to you guys to avoid it because right now we cannot really define it. No, I'm hearing some, I have some information about others taking screenshots of the questions that they will make use of the number of they will maximize the attempts in order to perfect the exam, things like that. They will give the questions, they will share it from not only within their class but also to another class. So with our setup right now, those things are possible. Those things are probably are happening. And my end, I cannot really control them. Okay, my what I can do is number one, I can have I will I did a while ago a moral plea to you. Na if you cheat, it's a lifetime taint on you, and you will always go back to it. No? Like if you become a successful doctor, Probably you will think about the time that you cheated. Think of it as a small thing, but not really, because being a medical doctor or being any professional will always have you'll always have your values as important grounding material, and such moments, no, of cheating are not really good things to remember. Okay. But on my end, if a report of a report of cheating comes to me, I'm able to discover it, I have enough evidence, I will, of course, make sure that the case will be endorsed to the Student Information Office. And I will make sure that the students or student involved will get their no, no, will get what they deserve for doing that. Okay? My consultation hours are not final yet. Okay, we haven't accomplished the loading of all the subjects, so I don't have any consultation hours as of the moment. But you can contact me anytime. I'll try to respond to you within 24 to 48 hours. Do not expect an immediate response. Okay, but during office hours, usually 10 a.m. to 4 5 p.m., I can reply real time. Or I will reply, definitely. I will reply real time. So, contact information, my work email, and my mobile number if you need to contact me for urgent matters. The references, the reading list are provided in the syllabus, so I just copy pasted them here. But primarily, our major reference, our major text is Hardin. So, this one. Uh, So we're going to use Hardin et al. Becker's World of the Cell. Okay. Um, all the chapters, the topics that I've discussed to you actually cover about 80% of the book. No? I've read this book 2, 3 times from cover to cover. So something that I think has helped me a lot, not only in teaching, but also in my uh, role as a graduate student. So you may have a copy. You may look for copies on your own. I suggest that you. I'm not sure if there are free copies, but if there are, then probably you can secure one for your own consumption. And if you have a copy, I will already tell you or advise you to do advanced readings, because everything will be lifted from this text, and then there will be side discussions or side material lifted from other reference materials. Okay, I think that's the end of my course orientation session. So make sure that you will be answering our transition survey para you can be automatically enrolled to your dedicated e-classes. And then for Module 1, please go over the netiquettes, the tips in succeeding distance learning, and go over the introduction activity. So I'll get to know some of you. As I think most of you pala, I haven't handled before. So I think half MBB 3.1 and MBB 3.2, I was not able to handle any of you during your second year. So MBB 3.3, somehow the faces or the students are very familiar to me. So please answer the self-introduction so I can on the weekend, I'll be able to read your outputs and try to remember some of you or most of you. Okay, so that's it.